My name is Cecilia Noecker, and the title of my talk is Using Metabolic Models to Integrate Microbiomes and Metabolomes. I'm a postdoc in the Turnbow Lab at UCSF, but most of what I'm going to talk about today is from my PhD work at the University of Washington in the Bornstein Lab. I'm broadly interested in the metabolism of the human gut microbiome. And this is an important area of research, in part just because of the enormous variety of metabolic routes by which our gut microbes can interact with host health and physiology. They can digest food, metabolize drugs, modify host compounds like bile acids and neurotransmitters, and produce complex secondary natural product metabolites. And many of these various pathways have been associated and linked to a wide variety of organ systems and diseases. And so for these reasons, an increasingly common study design in this area that I'll be focusing on for the rest of the talk is what I'm going to call a microbiome metabolome study. And what I mean by this is just any of this general class of studies where we have some population of subjects we're interested in, maybe with different phenotypes or disease status, and we profile their microbiome composition using metagenomic tools, and we also profile metabolites from the same subjects or samples using any of a variety of metabolomic technologies. And often the goal of this type of study design is to infer something about the relationships between microbes and their metabolites and how those interactions might ultimately affect host health. And so even though there were something like 700 studies of this type published in PubMed last year, there are still a lot of challenges in the analysis of these studies. We're pretty good at answering this first research question: what associations do we see between individual microbes, individual metabolites, and host features? But in many cases, we might be more interested in trying to answer more mechanistic questions, like do we see evidence that particular microbes are producing or using different metabolites, and if they might be doing so at different rates in different sample groups. And for instance, if we wanted to change the levels of a particular metabolite, what would be the microbes or the pathways that appear to be most responsible for determining the concentration of that compound? And the thing is that even though these are difficult questions to try to answer from a population study, we actually have a lot of useful information that we can contribute to answering these questions, or at least starting to better answer to better assess the evidence in support of them. So we have things like microbial reference genomes that are well annotated. We have pathway databases linking genes to metabolites that are regularly curated. And for some microbes, we have more and more reconstructions of the full network of metabolic reactions that they're capable of performing. And so this was a big goal of my dissertation work, was to develop analysis tools for analyzing paired microbiome metabolome data sets in the context of sort of reference information of what we know about microbes and metabolites, to evaluate what possible mechanisms might be at work in a particular data set. And so I'm going to describe to you the tool that ended up coming out of that work, Mimosa 2, which is now available online. But I first want to say a little bit about something that became really clear in the process of doing this methods development, which was that there's really not a clear framework for sort of evaluating the performance of this kind of integrative analysis method. And so first I had to define more quantitatively what exactly we might hope to achieve from analyzing microbiome metabolome data sets. And I also needed gold standard validation data sets where I would be able to evaluate and compare how well different options for analysis methods might achieve those goals. So to say a little bit more about that, first, so like I said, I needed to define more precisely the objective that we were hoping to infer from these microbiome metabolome data sets. And I think to have some intuition about this, I think it helps to think of a toy example where, let's say, we have one metabolite we're particularly interested in, and maybe there are 2 different microbial populations that are producing it and a third that can utilize it from the environment. And so maybe in some population of samples, they're all performing these processes at different rates in different samples. And so if we were to just come in and measure the amounts of each microbe and this metabolite in these samples, it would obviously be great if we could come up with a method to learn all of these detailed flux quantities, but that's maybe not totally feasible. And so we decided instead often what we might hope to learn is just what are the microbes, which microbes are sort of the most important drivers of ultimately differences in the concentration of a metabolite across different samples. And so we have a mathematical definition for this statistic. We call it key microbial contributors to metabolic variation. And so this is just a way to summarize the complex flux interactions between microbes and metabolites in terms of sort of how important is each microbe for each metabolite. So in this case, we might say the blue microbe has the largest impact on the observed concentrations of the metabolite, and so it's the key contributor. But to really define these key contributors, they are defined on the basis of sort of these reaction fluxes performed by each microorganism. And so to really have a sense of that ground truth, I needed a data set where we actually know all of those reaction fluxes for every microbe and every metabolite in the community. And so I ended up doing a lot of work with in silico simulated microbiome metabolome data sets that were generated using this framework developed by a couple other former members of the Borenstein Lab, Shun Chow and Colin, where basically what we're doing is predicting interactions between a population of microorganisms based on their metabolic networks, and iteratively updating those interactions for many time points. And you can run these simulations with sort of many different starting species compositions and many different environmental conditions to generate an artificial population of different microbial community samples. And so I did this for some analyses with a more artificial set of model 10 species communities, and I also worked with a more complex data set, simulated data set based on the species compositions observed in the Human Microbiome Project. And so in these detailed simulated microbiome metabolome data sets, I was able to evaluate how well different methods can recover these key contributor links between microbes and metabolites. And so one of the first things I looked at was whether just doing a simple correlation analysis between microbes and metabolites was sufficient to tell us about these mechanisms. So if we see that a microbe is highly correlated with a metabolite across many samples in a data set, how strongly does that tell us that this microbe is actually producing this compound? And actually what I found is that often correlations are not very informative. So for instance, in this simulation data set, we saw that significant species metabolic correlations contained many false positives where the microbe in question didn't even have any reactions relevant to the metabolite in question. It just happened to be highly correlated with it via an indirect association. And so I actually ended up doing a lot of analysis of how the interpretation of these associations is influenced by different characteristics of a study or a data set or of particular microbes and metabolites. And you can read about that in our M Systems paper that was published last year. But like I said, the main goal of putting together this evaluation and validation framework was to be able to develop new and better methods for evaluating relationships between microbes and metabolites. And so the tool that I ended up putting together, Mimosa 2, I'm going to tell you a little bit about how it works now. So Mimosa 2, like I said, takes as inputs a paired microbiome metabolome data set where you have some measurements of microbes across different samples and metabolites across different samples. And these can be generated using any number of different technologies for quantifying each of these features. And what Mimosa does with these data sets is first to construct a community metabolic model for each sample based on the microbiome data. So based on a number of different reference data options, how well does the tool, or what metabolic reactions does the tool think that the microbes in each sample are capable of performing? And from that, it calculates what we call these community metabolic potential scores that describe estimated effects of each sample and each taxon on different metabolites. And then we're able to compare those with the actual metabolite measurements. And if there's a strong statistical relationship observed, we can designate those as those metabolites as being potentially microbiome governed, meaning that the variation in metabolite levels is consistent with being explained by what we know about microbial metabolism. And for those metabolites, we can then go ahead and decompose how much the metabolic potential scores and therefore the metabolites can be explained by these different microbial taxa, i.e. these key microbial contributors we're trying to directly infer. And so these are the two main outputs of the tool. And so like I said, I validated this in multiple simulated data sets, showing results from 2 of them here, that a couple of different model fitting options for Mimosa 2 achieve higher precision and specificity than correlation-based methods, even ones that incorporate some metabolic network information. I also evaluated Mimosa 2 in using data from an experimental synthetic community study of the honeybee microbiota. So what these researchers did is they profiled these honeybees colonized with this 7-member community. And so we analyzed that data with Mimosa 2, and it inferred different expected contributions by each of these strains to each of these metabolites, which is what's shown in the heat map here. But they also had honeybees that were colonized with just each of these strains individually monocolonized. And so we compared the metabolite changes observed in monocolonization with the community effects and found that many of the contributions inferred by Mimosa 2 are also supported by these monocolonization studies. So Mimosa 2 is available online now at that URL. You can access a Shiny web application for running it that also generates some interactive results summaries. It's available as an R package to integrate into analysis pipelines, and we have some of the pre-processed reference databases available for download as well. And so, as I've told you about here, I've described frameworks for evaluating methods to infer metabolic mechanisms relating microbes and metabolites from population survey studies, and in particular a new tool, Mimosa 2, for doing this that's been validated in simulated and synthetic data. I do want to highlight that an important limitation of Mimosa 2 is that since it's using this reference information, it can only make inferences about the microbes and metabolites for which we have good reference information, which obviously leaves out many important parts of microbial metabolism. And so that's one of the things that I'm working on now in my postdoc, is developing strategies to combine metabolic modeling with careful experiments and metabolomics data in controlled environments to be able to fill in some of the reference gaps for understudied gut taxa of interest. And so I think there's going to be a lot of continued new insights and opportunities for improvement of these databases and these methods going forward. And with that, I'd just like to acknowledge my past and current labs and past and current funding. I also really want to thank the organizers for giving me the opportunity to present here today. I am very happy to take questions at this time, and if you're interested in using or checking out Mimosa 2 further, this is the URL for the documentation website. So thank you very much.